Some of the characters are of living things and others are of non-living things. So let's move to the next today, the types of viruses. Now we can categorize viruses into three basic categories: animal viruses, plant viruses, and bacteriophage virus. Okay, animal viruses are those viruses which infect animals. Plant viruses are those viruses which infect plants. Bacteriophage virus is that virus that infect bacteria. So virus can infect as small organisms as bacteria as well. They can infect plants as well, and they can infect animals as well. So these are the three basic types of viruses. Then there are subtypes in animals virus and plant virus. Definitely there are subtypes as well, but the basic types are these three. So they are infecting animals, they are infecting plants, and they are infecting bacteria as well. Okay, now let's see a little bit about viral diseases. We are not going to discuss here in detail. The diseases we will discuss in functional biology, that is from chapter number 9 and onwards. So here I am just giving you a little bit of idea. You are not supposed to go very deeply to this one, okay? Because there is no deep questions here. There is no deep explanations here. We will discuss that later in the chapters that comes in chapter number 9 and onward. So we'll discuss that there. So let's see poliomyelitis. This the disease, we call it polio disease, okay? So it is caused by a virus whose name is poliovirus, okay? Then we are having colds. Colds are basically of very many types, okay? You have seen a kind of cold today's coronavirus as well. It's also a sinus virus. It causes infection in respiratory tract. So colds have almost more than 200 viruses that have been identified till today, and definitely more than that can occur in the future as well. Their DNA, RNA sequence will change, and their types can be changed, okay? Then we are having measles and mumps virus. It was caused by measles and mumps virus, okay? It caused a disease called measles and mumps in children. There's basically a smaller, you can say, fever in the body, okay? And then along with that, there are kind of small pimples on the body having pus in that, okay? So they are called measles and mumps. Then we're having rabies. Rabies is basically present in the saliva of infected dogs. If any dog bites you, it can transfer rabies to your body, okay? And if rabies is transferred to your body, the person he started to afraid from water. So it's a psychological condition in which you are afraid of water. You cannot drink water, you cannot face water, and ultimately psychologically the person is that much disturbed that he become mental, and that the person become die. Then we are having AIDS, acquired immune deficiency syndrome, that is caused by HIV, and HIV was discovered in the air in 1985. So some of the diseases we have discussed here, not many of the diseases, but there are some of the diseases, and there is no explanation over here right now. So we will discuss them. I'm telling you people time and again, we will discuss them in chapter number 9, 10, 11, 12 and onward. All the chapters we are having these diseases, we will discuss all the diseases over there in detail. Now let's see how the viral diseases are spread. How are the diseases spread? First of all, the viral diseases are spread by airborne droplets. Okay, if somebody has the virus and he sneezes and cough, so it can be transferred to the other people who are basically present with him in the surroundings. So airborne droplets, the virus can spread through sneezing and coughing. Okay, the virus can be present in contaminated water as well. Then the virus can be present and it can be spread through contaminated food as well. Okay, by the blood and other body fluids as well. Like for example, you transfuse the blood, so you can transfer the blood, and if the blood has virus, it can be transferred to your body as well. So then we are having the use of non-sterile surgical instruments. Like for example, we are using the instrument for the operation. Okay, we are using the instrument for the teeth and many other things. If they are not sterile, they are not clean, so that can cause viruses spread in your body. And if you are using a syringe which is not clean, which are used, so there are chances that you can get the virus into your body. Okay, how can we control the viral diseases? What are the control of the viral diseases? The first thing, the most important thing about the control of the viral disease is by maintaining proper hygienic condition, cleanliness around your environment, cleanliness of surfaces, cleanliness of your hands, your face, your body. This is called personal hygiene and hygiene of your environment. So if your environment is clean, you are personally clean, virus is not going to infect you. But if your environment is not clean, your food is not clean, your water is not clean, your hands are not clean, then the virus can easily enter your body. By proper vaccination, okay, this is one of the method, one of the technique by which we can take a pre-precautionary measure so that we cannot face the virus in the future, or the virus should not enter to our body, or if it enters our body, there are antibodies present in our body which can attack the virus and can cancel its effect as well. Okay, and then we can spread awareness in the people, the educated people. We are the educated people. If we know about something, we should spread the awareness. If we are not spreading the awareness and people are doing what they are doing and they don't know about how to take care of themselves, then the virus can affect their bodies. So these are the control ways by which we can control a virus. Okay, let's come back toward the screen if there is any question regarding the virus. But as I told you, we will discuss diseases in detail as well. Barira, daily I am sharing the slides. So what is the problem? I'm sharing the lecture, I'm sharing the problem. Sorry, I'm sharing you the slides as well. So if there is any problem, you can ask them.
Sir,
Yes.
sir, what are airborne droplets?
Airborne droplets mean the droplets that can carry in the air. Like for example, when you sneeze and you cough, so the particles which is throwing out of your mouth, they can travel in the air. And when they travel in the air, they can go and they can enter to the body of any other person. So that cause the virus to spread.
Sir, in AIDS you said HIV. What is HIV?
HIV is a human immunodeficiency virus. It causes a problem in your immune system. How can virus infect bacteria? Virus is much smaller in size than bacteria. It can enter to the body of bacteria and they can replicate in the body of bacteria and then destroy the body of bacteria.
Sir, what is meant by contaminated?
Sorry?
What is meant by contaminated?
Contaminated mean ganda in simple word. Having contamination, having microorganisms, having bacteria, having fungus, having viruses in them. That is called contaminated.
Okay, thank you, sir.
सर अगर वायरस जो है वो बैक्टीरिया को इन्फेक्ट करता है तो उससे कोई डिजीज हो सकता है इंसान को?
It's not like viruses are very much specific. Okay, they're not going to attack each and every body. Viruses have very specific relationship. Like for example, we discussed some of the viruses. Okay, we discussed that there is a virus called poliovirus. So what does the poliovirus do? It causes only one disease in your body, that is polio disease, poliomyelitis. And what is the disease of poliomyelitis? It is the disease of your nervous system. So it enters your digestive system and from there it goes to your nervous system. It causes only disease in your nervous system. It cannot cause disease in any other part. Similarly, bacteriophage virus that infect bacteria that cannot infect human being. Okay, so if like for example we are having hepatitis virus, so it is going to just infect your liver and cause hepatitis to you. It cannot cause any other kind of disease. So viruses are very much specific. They are not attacking the whole body. They are attacking limited parts of the body. Clear?
Sir?
Okay, sir.
Any other? Heather Imam, I will share the slides. I will share the lecture. Don't worry. Okay, any other question?
Bacteria do help, like there are some good bacteria as well, which
We will
like
discuss that. Now, the next topic we are having bacteria, okay? We will discuss that.
ऐसे सर हमारे काम में वायरस आ सकते हैं जैसे कि बैक्टीरिया काम में आ जाते हैं?
बिल्कुल भी नहीं. Viruses mean poison. Okay, they mean poison. So it is only going to harm you. It is not providing you any kind of benefit.
सर वही क्वेश्चन मैं पूछ रही थी, आपने कंप्लीट नहीं करने दिया मुझे.
Ask the question.
वो कर लिया ना सर सकीना ने.
Okay, sorry then. If you have any other question, you can ask. Any other question?
No sir.
Okay. So let me tell you some secret about viruses. Viruses are basically the organisms which are very much weak. Okay, they are very weak organisms. So when they enter your body, at once they are not going to show their presence in your body. Okay, because they're weak. You have an immune system that show resistance against the diseases. So if there is a smaller thing, like for example a weak thing, come to your body, your immune system will respond and it will catch those things. So viruses do what? Viruses go to a specific organ. They go to that specific organ and they start reproducing in that specific organ. So when they are many in number, means they are much more in number and they are much stronger in number, then they start infection. So before that, they do not start infection. They remain inside, calm and quiet. And after that, when they are more in number, they start their infection because then they are more in number. They can reproduce themselves, they can replicate themselves, and then they can attack you. So they are just like the organisms who are not going to mess with you in the beginning. But when they become more in number, then they start messing with your body. Okay? Am I clear to you now?
Yes sir.
Good. So this was the topic of virus. We have completed the topic of virus.
sir.
Now we are moving to the next topic, that is of bacteria. So let's again start with the next topic. We are having the next topic we have is bacteria. The person you are seeing in the picture is known as Anthony van Leeuwenhoek. He is the person who studied and discovered bacteria first of all. And in the next picture, you can see a device. This device is known as Van Leeuwenhoek microscope. It is Anthony's Van Leeuwenhoek microscope. So he was basically a microbiologist, and he was working in the British Royal Society, and he was serving them and making for them beverages and alcohols. So in that beverages and alcohols, he find out the presence of bacteria with the help of the microscope, which you can see in the diagram. It is a very simple microscope. Okay, initially this microscope was discovered. After that, many kinds of microscopes has been discovered, and we know about them as well today. So let's see what bacteria are. The discovery of bacteria. So bacteria are basically considered the smallest and simplest unicellular microscopic prokaryotic organism. Remember, bacteria are smallest, simplest, unicellular, microscopic, and prokaryotic. It means bacteria come under the group of prokaryotes. Okay, kingdom prokaryota. They come in the kingdom prokaryota. So they are the simplest unicellular organisms present in the world. They were discovered first of all by Anthony van Leeuwenhoek. Okay, and in the year 1676, and he discovered that from the mouth of the human being. The scrubs present in our mouth, he took those scrubs and saw that under microscope and found out that living thing that is known as bacteria. So the bacteria which he discovered was the genus of Pseudomonas. The Pseudomonas was basically a bacteria which is a crescent-shaped bacteria. And what is the crescent-shaped bacteria? The crescent-shaped bacteria mean the bacteria like a moon, a crescent moon. So this was a curved bacteria, okay, which he found out in the mouth of the human beings. And after that, the study of bacteria started. So this was about the discovery of bacteria. Again, bacteria are considered as the smallest, simplest unicellular prokaryotic organisms. Okay, they were discovered first of all by Antony van Leeuwenhoek in the year 1676. And where he discovered them? He discovered bacteria in the mouth of human beings. The first species which he discovered is Selimomonas. Okay, Selenomonas. That is the bacteria that is known as crescent shape. And crescent shape means a bacteria which is basically a moon-shaped bacteria. So it is a curved moon-shaped bacteria. Initially you see the moon, not a full moon, the crescent moon. That crescent moon is basically the shape of the bacteria which he discovered. Okay, then let's see the characteristics of bacteria. What are the characters of bacteria? Again, this is very much important questions. Okay, the first one, let's see the habitat. Habitat is basically the place of living. So where do a bacteria live? Bacteria grow in every habitat under all possible environmental conditions on the earth. What does it mean? It means that bacteria can survive in all the habitats of the world. It can survive in freezing environment. It can survive in boiling environment. It can survive in oceans. It can survive in the air. It can survive in the fridge. It can survive everywhere in the earth. So all the possible habitat, whatever habitat is, bacteria can grow over there. Okay, it is possible for the bacteria to grow over there. Let's see. Bacteria are basically the largest number of creatures. Okay, mean the number of bacteria present is largest than any other creature. And the reason behind that is because bacteria reproduce in 120 minutes. Okay, and each 120 minutes bacteria can reproduce itself. So when they reproduce themselves time and again, in one day they will reproduce many times. Okay, they will reproduce many times. And when they have reproduced many times, they are going to have many offsprings in them. So their number of offspring are much more as compared to any other organism. Okay, their survival. Bacteria can survive in each and every temperature. If the temperature is freezing, they will survive. If the temperature is hot as the sulfur spring, they can survive as well. For example, in volcano, they can survive in the volcano as well, and they can survive in the Antarctic Ocean as well. So everywhere they are possible to have survived, and they can survive everywhere. What is the size of bacteria? A bacterial cell has 0.2 to 2 microns in width and 2 to 10 microns in length. Okay, its width can be 0.2 to 2 microns, and its width can be 2 to 10 microns. Microns are a little bit bigger than nanometers. Okay, last about viruses we studied about nanometer. Nanometer is a smaller unit. Microns or micrometer are a little bit bigger unit. So bacteria are bigger in size than viruses. Their cell ranges from 0.2 to 2 microns in width and 2 to 10 microns in length. Okay, now if you have any question in the characteristics of bacteria until now, so you can ask, then we can move on. Anybody is having any question until now?
सर
Yes, ask the question.
सर जो आपने बताया कि बैक्टीरिया सरवाइव कर सकता है हॉट टेम्परेचर में भी और कोल्ड टेम्परेचर में भी, तो सर वायरस सरवाइव कर सकता है ऐसे ही बैक्टीरिया की तरह?
Virus is not that much virulent. Okay, virus is basically they can survive in specific areas at specific temperatures. Virus cannot survive everywhere because they are particles. Particles, they are not living things that will be present on the surfaces. If they are present on a surface, it's non-living. It cannot behave like bacteria. Bacteria is a true living organism.
Okay, sir. Thank you.
सर
Yes.
सर जो बैक्टीरिया है वो हर उसमें जैसे फ्रीजिंग और हॉट वेदर में भी रह सकता है, तो सर फिर हम अपनी जैसे खाना या प्रिजर्व जो हम करते हैं वो हम रेफ्रिजरेटर में फिर क्यों रखते हैं?
We keep it because the growth can be preserved for some time. Means you can stop the growth for some time. You cannot you cannot kill the bacteria, okay? Means they are not going to be killed, but they will be freezed. The growth will be stopped. Like for example, if you put something in the freezer and you put it out of the freezer for some time, what happens? It again shows the contamination. Why show the contamination? Because the bacteria are present over there. Okay, the bacteria are present, but they were inactive. So as it comes out, it became active again and start their work again. The reproduction are just stopped there.
तो सर हम ये कह सकते हैं कि बैक्टीरिया कोल्ड उसमें ज़्यादा मतलब इनएक्टिव होता है जबकि हॉट वेदर में.
Let me tell you people, the microorganisms are normally more active in moderate environment. If the temperature is moderate, the microorganisms scale up growth and infection is very much more. But if the temperature is on the hotter side and temperature is on the very colder side, their infections are very less then, because that is not a favorable condition then. Okay, only the favorable conditions are basically those which is the moderate environment. Heather Imam, there are lots of techniques. Definitely we have electron microscope, we have smaller microscopes. With the help of that, and we have devices through which we can measure the smallest of the objects as well. We have micrometer, screw gauge, we have vernier clippers, we have more other things through which we can normally measure them. Arisha, sulfur spring mean volcano, the hot volcano. And you know that anything's come in its way, they are burnt down, but bacteria can still survive over there. Done? Okay, now we are moving to the next topic, that is the structure of bacteria. Let's see what the structure of bacteria is. The structure of bacteria. First of all, the cell wall. Bacteria is a cell, and it has a cell wall in them as well. It has a proper shape, okay? So bacteria are protected by a rigid cell wall. A rigid mean that is firm, sakht, okay? So that is a rigid cell wall, which is different from the cell wall of the plant. Plant cell wall has cellulose, but bacterial cell wall do not have cellulose. Bacterial cell wall is made up of peptidoglycan. Okay, peptidoglycan. Peptide mean protein, glycan mean carbohydrate. So they are made from two things. One is protein, second is carbohydrate. From the combination of protein and carbohydrate, the cell wall of bacteria is formed. Then the cell membrane. Next to the cell wall, the first layer is the cell wall, the second layer is the cell membrane, which is present in all the cells. Okay, so after the cell wall, we are having the cell membrane. A cell membrane has many pores in it, okay, which is basically made up of lipoprotein. Lipo means lipids, fats, and protein, you know that they are the compounds are made from amino acids. So the cell membrane is made up of lipoproteins. The cell wall is made up of peptidoglycan. Peptidoglycan means protein plus glycan mean carbohydrate. Lipoprotein means fats plus protein. So from these molecules, their cell wall and cell membrane are made. The cell wall is also used for the function of respiration, because in their cell, the mitochondria is absent. And you know mitochondria are present in eukaryotic cells. They are used for respiration. So when there is no mitochondria in the bacterial cell, they use their cell membrane for the process of respiration. Okay, let's move on. Cytoplasm. Inside the cell membrane, all the part is known as cytoplasm. It is a jelly-like substance, and it contains vacuoles, ribosomes, and glycogen particles. Okay, vacuoles are basically the particles in which they can store their food and they can remove their food out. The ribosomes can make protein for them, and glycogen particles are basically the particles of carbohydrates present in them which they can use for their food. So these are the different particles present inside the bacterial cell. It is a very simple cell. It do not contain the particles or the organelles which are present in the eukaryotic cell. In the chapter number 2, we will discuss about all these particles like endoplasmic reticulum, plastids, mitochondria, ribosomes, vacuoles, all we will discuss in cell in detail. But in bacterial cell, there is no endoplasmic reticulum, there is no plastid, there is no mitochondria. it means that they do not have any kind of complex structure in them. Okay, then there come nucleus. A bacterial cell do not have a well-organized nucleus. As we discussed that they are prokaryotic. So prokaryotic means that the nucleus is not true. The nucleus is not well-organized. Okay, they only have a nuclear material or chromosome present in them. In the cell, only chromosomes are present which behave as their nucleus, but the true nucleus is not present in the cell of the bacteria. Okay, then we are having flagella. Flagella is basically an organ which is present in the majority of the bacteria for the process of movement, mobility. So with the help of the flagella, bacteria can move from one place to another. Okay, with the help of the flagella, bacteria can move from one place to another. Okay, they are very thin structures and they are attached to a cell wall. Some bacteria have one flagella, other have many flagella. So, and some of them have no flagella. So the bacteria who do not have the flagella, they cannot move. And the bacteria who contain flagella, they can move. Then we are having pili. Pili are basically hair-like structures which are attached to the cell of the bacteria. And they are basically involved in the conjugation of bacteria, means they help in the reproduction of bacteria. Conjugation means reproduction. When two bacteria combine together and they reproduce, so this process is known as conjugation. So pili help them to join together and have the process of conjugation or reproduction. So this is the structure of bacteria. Let's see the diagram. In the diagram, you can see that there is a cell wall present. Okay, outside the cell wall, an extra layer of capsule can be present in some of the bacteria. In all of the bacteria, it's not present, but in some it can be present. Okay, then we have the cell wall. Cell wall is actually the first layer. After the cell wall, we are having the cell membrane. Okay, inside the cell membrane, the whole part is known as cytoplasm. And inside the cytoplasm, we are having the DNA present, the chromosome present. It is not a proper nucleus in them. Okay, then the ribosomes are present in them. What? Okay, and the flagellum is present in them. So this is the diagram of a bacteria. You can see the diagram of a bacteria. A bacteria has a proper shape. This is a rod-shaped bacteria. This bacteria is a rod-shaped bacteria. Other bacteria can be round, other can be curved, and many other types as well. And these are pili. These structures are known as pili. Okay, a singular is known as pilus, and many of them are known as pili. Let's see back to the screen. If you have any question now, you can ask it. Sayyid, Ali Sayyid, virus से भी छोटे organism अब discover हो चुके हैं, but we are not going to discuss them over there. Prions and viroids are discovered, which are smaller than the viruses as well. Okay, peptidoglycan. Arisha, peptide mean protein. Peptide mean protein. A glycan mean carbohydrate. Peptidoglycan is a compound which is made up of protein and carbohydrates. And ribosomes are basically the smallest particles present in the cell. They are responsible for making up proteins. They make proteins for the cell. Yes, any other question? If anybody is having any kind of question, so you can ask it right now. We have discussed the structure of bacteria. We have discussed the characteristics of bacteria. And we also have discussed about the diseases of the viruses and its spread and control. So if there is any problem, you can ask the question. What is meant by true nucleus? Sayyida Zoha, true nucleus means a nucleus which contains nuclear membrane. So if a nucleus contains nuclear membrane, it is called true nucleus. And if a nucleus does not contain a true nuclear membrane, then that cell is known as prokaryotic cell or false cell.
Sir,
Cell
can you
wall.
again explain what is pilus?
I'm explaining it. Okay. First of all, cell wall. Cell wall is basically a structure that is present in some of the cells. Okay. So in bacterial cell, cell wall is present. In plant cell, cell wall is present. In fungus cell, cell wall is present. Cell wall जिन भी cells के अंदर present होता है, उस cell का एक proper shape होता है. Okay. The cell which do not contain cell wall, they do not have a proper shape in them. Okay, pilus is basically structures. They're hair-like structures present in the bacteria. Somewhere it can be present around the cell, or sometime it can be present at one place as well. They are used to help bacteria in the process of reproduction. Any other? Any other question? I think there is no question. All is clear.
Yes, sir.
Very good. I'm stopping the recording now.